Professor Dr. Jennifer Ann Harikrishna from the Department of Genetics, Faculty of Science, University of Malaya. The lecture will be commencing shortly. For your information, today's inaugural lecture is organized for fellow professors in University of Malaya to share their knowledge and insights in their respective fields. It also complements the promotional process and service extensions for professors in University of Malaya. To ensure that today's inaugural lecture runs smoothly, I would like to highlight a few house rules before we begin. Firstly, please stand up when the arrival of the VIPs is announced and remain standing thereafter for the national anthem and the UM song. All guests are kindly asked to remain in the auditorium until the end of the lecture so as not to disrupt the speaker. And lastly, I would like to remind everyone and myself to switch off our mobile phones or set them to silent mode. Your cooperation is much appreciated. Thank you very much. Saya takut tekan-tekan. Okey. Okey. Oh, untuk orang pendek ni. Oh, okey. Prof tinggi. Prof tinggi. Tinggi tak payah, nanti akan masukkan lagi. Okey. Kita boleh punya. Tak payahlah. Tinggi je, tak payah. Tak payah. That's really good. Hey, Announcing the arrival of Yang Berbahagia, Professor Dr. Rofina Yasmin Othman, Associate Vice Chancellor, Industry and Community Networks, University of Malaya, and Professor Dr. Jennifer Ann Harikrishna. Please remain standing.
Malaya, kebanggaan kita semua, bertambah para ilmuwan di berbagai lapangan. Kami bertekad di hati mencapai cita mudi, tradisi kecemarahan menepati wawasan. Di sini kami sedang berbakti, terus berinovasi. Kami harapkan generasi muda, cerdas nusa dan bangsa. Kasih kesayanganku, sungguh banyak jasamu membentuk insan mulia, penuh berbudaya. Di sini kami sedang berbakti, terus berinovasi. Kami harapkan generasi muda, cerdas nusa dan bangsa. Pasifik kesayanganku, sungguh banyak jasamu membentuk insan mulia, penuh berbudaya.
Ladies and gentlemen, thank you, and please be seated. Yang Berbahagia, Professor Dr. Rofina Yasmin Othman, Associate Vice Chancellor, Industry and Community Networks, University of Malaya. Yang Berbahagia, Professor Dr. Shaliza Ibrahim, Associate Vice Chancellor, Research and Innovation, University of Malaya. Yang Berbahagia, Tan Sri Emeritus Professor Datuk Dr. Augustine Ong Soon Hock. Yang Berbahagia, Tan Sri Datuk Sri Dr. Saleh Muhammad Noor. Yang Berbahagia, Professor Dr. Helen Nair, Fellows of the Academy of Science, Malaysia. Yang Berbahagia, Professor Dr. Zanariah Abdullah, Dean, Faculty of Science. Yang Berbahagia, Associate Professor Dr. Nurhayati Zainal Abidin, Head, Institute of Biological Sciences, Faculty of Science. Tan Sri's Dato' Datin, distinguished guests, management of University of Malaya, fellow professors, colleagues, ladies and gentlemen, Assalamualaikum and a very good afternoon. Welcome to the inaugural lecture by Professor Dr. Jennifer Ann Harikrishna, entitled It's a Small World After All, From Microbiology to MicroRNA. Without further delay, I would like to invite the chairman for today's inaugural lecture, Yang Berbahagia Professor Dr. Rofina Yasmin Othman, Associate Vice Chancellor, Industry and Community Networks, University of Malaya, to take over the floor. Please welcome.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Yang berusaha Profesor Shaliza Ibrahim, Associate Vice Chancellor, Research and Innovation. Yang bahagia Tan Sri Emeritus Profesor Datuk Dr. Agustin Ong Sung Ho. Yang bahagia Tan Sri Datuk Sri Dr. Saleh Muhammad Noor, Fellows of the Academy of Science Malaysia. Yang berbahagia Profesor Dr. Zanaria Abdullah, Dekan Faculty Science, Universiti Malaya. Yang berusaha Associate Professor Dr. Nur Hayati Zainal Abidin, Ketua Institute Science Biology, Faculty Science, Universiti Malaya. Tan Sri, Datuk-Datuk, Datin-Datin, Distinguished Guests, Pengurusan Universiti Malaya, Profesor-Profesor, pelajar-pelajar yang dikasihi, tuan-tuan dan puan-puan sekalian. Syukur kehadirat Ilahi pada hari ini kita dapat bersama pada hari ini yang kita dapat bersama pada hari yang bermakna ini untuk bersama-sama mendengar syarahan perdana Profesor Dr. Jennifer Harikrishna. Saya bagi pihak Universiti Malaya mengalu-alukan kehadiran tuan-puan ke Universiti Malaya dan amat terharu dengan kehadiran yang ramai pada hari ini untuk mendengar syarahan beliau. Ladies and gentlemen, allow me to continue my small speech in English. It's a small world after all, from microbiology to MicroRNA, and this is such a meaningful title to me particularly. It is my great pleasure and my great honor to be given this task of introducing Professor Dr. Jennifer Harikrishna, Institute of Biological Science, Faculty of Science, and in delivering her inaugural lecture. Let me introduce, in a small way, Professor Jennifer Harikrishna. So Jennifer N. Harikrishna, or as I used to know her as Jennifer N. Kirkham, was born in the city of Nottingham, United Kingdom, as the second child and only daughter of a staff nurse and a mechanical engineer. And she was a little kampung girl, brought up in a Nottinghamshire village close to the River Trent. She was not just any schoolgirl. She was an extremely active young lady, winning prizes for music, for academic achievement, and if you don't know it, she was an athlete, school champion for 200 meters and a high jump, a girl guide, a Duke of Edinburgh Award winner, and a member of Nottinghamshire Country Youth Orchestra and Concert Band. Surely a really good sign of things to come. She went on then to do a Bachelor of Science degree in Microbiology in the University of Surrey, UK. And this is where our history started. And it's a small world, definitely. Jennifer and I, just to share with you, met on a train. Two girls, scared, first time out of school, on the way to interview. We were both on the same train for an interview at the University of Surrey. And we sat opposite each other and said, "Where are you going?" "Oh, I'm going to Surrey for my interview." And there it was. Many years ago, and I don't want to say how many years ago, but we were 19. We were 19-year-old girls on the way to an interview in the University of Surrey for BSc Microbiology. And that was when I first met her. And our history has since been intertwined off and on, and here we are today. So the Bachelor of Science degree in Microbiology at the University of Surrey, don't be deceived by her demeanor as the pure English rose. At the University of Surrey, she was an active Aikido expert. Okay, high kicks and big sticks, you know. And that was the hallmark of Jennifer, very determined in everything that she did. And certainly she excelled at the university. She was also a member of the University of Surrey Morris Dancing Club band. So again, this is a diversity of the talent of Prof. Jennifer. Completed her industrial placement, and then during the tenure that she was in University of Surrey, she made her first visit to Malaysia. 1982, she stepped foot in our soil, and I don't think she's ever looked back. It has kept her coming back to our country, and we're so happy that she's now very much a part of Malaysia. On graduation, she was awarded a Whitbread Scholarship for PhD studies, and she went on to do her doctoral thesis on molecular genetics and industrial yeast at the Cranfield Institute of Technology, Cranfield University, UK. And there again, she excelled beyond expectations and was awarded the Chancellor's Gold Medal for the most outstanding graduate student of the year in 1990. But that's not all she picked up there. As you know, when she was in Cranfield, I think she was also very fortunate, or unfortunate, I don't know, you tell me, to have met a young man by the name of Harikrishna Singham, who went on, of course, to be her partner in life. And I think she'll definitely tell you a lot more about that story in her talk. And so this followed on with a 2-year postdoctoral fellowship at the University of California in San Francisco, a very, very prestigious postdoctoral fellowship indeed, before moving on to Malaysia in 1989 to 1991. And after 2 short postdoctoral fellowships at the University of Malaya, so as you can see, her first foray into Malaysian academic life was in University of Malaya in 1989. So she has a very, very long and strong history of service to the university. She then joined as a pioneer with, in fact, a very innovative and pioneering company, Trop Bio Research Sdn Bhd, the founder of whom is here today, Tan Sri Datuk Sri Saleh, a company which was ahead of its time, and joined as the head of the molecular biology group, where she remained for about 5 years, 1996 and 2001. So Prof. Jennifer then expanded from beyond academia to having great industry experience. And because of that, she was then lured back into academia and joined as associate professor at the University Putra Malaysia from 2001 to 2002. During this period, she was also awarded visiting scientist at MIT for a national program that was funded by MOSTI. So during this time as well, Professor Jennifer continued to pioneer new and exciting ventures in Malaysia, and she was among the pioneer lecturers at a new graduate university, the Malaysia University of Science and Technology, MUS, where she spearheaded the molecular biology program from 2002 to 2006. And in this program, she taught the MSc Biotech program and supervised M-Biotech thesis, and started collaborations then with the University of Malaya. So she cannot run away from the University of Malaya. I think this was her calling from the beginning. So finally she joined the University of Malaya as an associate professor in April of 2006. Interestingly, all this time, I didn't know that she was in Malaysia. I was, of course, doing my graduate studies abroad. And it was just like fate, I guess, that our paths met again, and we meet again and again, and finally we became colleagues in the University of Malaya. So she was the program coordinator for the BSc Genetics and Molecular Biology program from 2007 to 2009, and more recently was appointed as head of the Center for Research in Biotechnology for Agriculture in 2009, and its director when SEBA received UMCOE status in 2013. And I have to say that Jennifer has been instrumental in keeping SEBA in the forefront of the Center of Excellence, and we're on our way to hopefully becoming a National Center of Excellence in Biotechnology under her stewardship. Prof. Jennifer's contract as a full professor at the ISB, or the Institute of Biological Sciences, Faculty of Science, began in August 2011, and currently she holds dual appointments, both as a professor in the Faculty of Science as well as director of SEBA, University of Malaya, which actually highlights how much faith the university has in her ability to lead. Prof. Jennifer has a World of Science H index of 10 with an average of 16 citations per paper. She has supervised more than 40 graduate students, with 8 of whom have graduated with PhDs. She's the author of more than 50 full-length peer-reviewed high-impact papers, has time to do patents, copyrights, and commercial activities as well. A true all-rounder. She has been a life member of Persatuan Genetics Malaysia since 1992, a life member and a very active one of the Malaysian Society of Malaysian Molecular Biology and Biotechnology, being on the committee from 22 to 2016, treasurer from 23 to 27, and she has a string of awards to her name in research and innovation, gold medals, silver medals, bronze medals, you name it, starting from her time in Cranfield University when she won the Lord King's Northern Gold Medal, right until today when she continues to win medals for her innovation and research. She's also handling editor for several journals, including the very prestigious Annals of Botany since 2015, and been a reviewer for high-ranking journal, placed in the 86th percentile for verified reviews and 86th percentile for editorial contribution to World of Science journals. So, Prof. Jennifer, certainly that doesn't do justice to all the science and innovation that you have brought into our lives. It doesn't do justice to all the students that you have nurtured and supervised all these years. And it remains for me to invite you to present your inaugural lecture, It's a Small World After All. Professor Jennifer N. Harikrishna.
Okay. Good afternoon, everyone. Selamat petang, selamat sejahtera. And thank you very much, all of you, for joining me here. And I'm also saying that to many friends and family who are watching on YouTube, I believe, as this is broadcast. So thank you all for joining me. Thank you also, Prof. Yasmin, my great friend and collaborator, and certainly a big part of my story, for that very kind introduction. When I was trying to come up with themes for my talk, I decided that A Small World was suitable, as Yasmin has mentioned, keep bumping into the same people all the time, and amazing how often you meet someone and you start talking to them about someone else and they know each other. And I think that's particularly true in scientific research, because as we become more and more specialized, we realize that we end up being a group of very small people around the world who actually have a strong interest in a particular subject. So this is one of the reasons that I say that one of my themes then is the small world, the connections, the collaborations, and the specialization. Another theme that will be in today's presentation is abiotic stress. And this also falls under the fact that it's a small world. In fact, it's our only world. We all have to live on this one planet, and sustainability and use of sustainable and renewable resources, things like food security, are so very important. We're also facing climate change. We're facing dwindling resources for land. And so one of my areas of research has been focusing on abiotic stress. This is stress coming from non-biological, non-disease sources. And climate change, I think, is a big part of that, even though maybe there is some human or biological component within there. And the third area, because that was in the title, the small RNA or the microRNA, that's because the year I joined University of Malaya as an associate professor in 2006, this was really a field that was starting to explode. Small RNAs were beginning to be really understood for their importance in genetic regulation, and that area fascinated me. I was looking for things to work on that other people were not working on, and not many people were looking at that yet in tropical crops. So, as Yasmin has said, we started out, the pair of us, as microbiologists. And microbiology is the study of really, really small organisms. So small, again. And despite the fact that they are so very small, they have really, really big impacts. And we know this best from what we consider bad microbes, things that have caused diseases, even pandemics. And they've also caused famines and crop losses. And these have been such big impacts that they've changed human history. But at the same time, we have good microbes. And in fact, the good microbes far outweigh the bad ones. So for all of human history, you'll find populations have been actually using microbes, doing biotechnology, whether they knew it or not, for food preservation. And some good examples in Malaysia would be tapai and tempeh, which involve microbial fermentations. They also don't only cause problems to us. Microbes fight with each other. They make nasty toxins to try and kill one another to get a competitive advantage. And because of this, we can use those toxins, and we call them antibiotics, and they can be used to treat human diseases and animal diseases and have saved millions of lives. We also use microbes against themselves when we develop them into vaccines, also having saved millions of lives. So you can see that there's a lot of good things coming out of microbes. What maybe people who don't work in plants don't realize is that we also use these genes to protect crops. We use the genes from microbes, or the whole microbe themselves, in the case of Bacillus thuringiensis, or Bt toxin, to protect crops. And the final thing that's, to me, really important about microbes is that they are such fantastic model systems for molecular genetic studies, which is my area of interest. And in this role, they were really the forerunner of understanding the roles of RNA and the different ways that RNA is behaving in cells. And so RNA-based regulation would eventually lead to the discovery of microRNAs. So today, please come with me on a journey from English Kampung girl through to Kampung University Malaya, where we are today. And I do feel often that University Malaya is a lot like a kampung. Everybody knows each other. We're always poking our nose in each other's business, want to know what's going on. And that's a really lovely thing about being here. This photo is of me and my mum out picking flowers in the countryside. As Yasmin mentioned, my mum is a nurse and my dad's an engineer. They're both really keen gardeners, avid bird watchers. They love nature. I was dragged around the whole of UK on camping holidays. And so I don't think it's really such a surprise that I ended up as a biologist. And as Yasmin also has said, I promised to embarrass her, a sweet young thing of 19. And yes, it's true, we met on a train. And we were actually the only two people who had interviewed on the same day and ended up on the course. We became lab partners, project partners, and spent a lot of time together. And she did invite me and her family most kindly during the first summer holidays at the end of our first year of university to come out and visit Malaysia. And she'd made it sound such an amazing place that I really wanted to visit, and it didn't disappoint. And her family are wonderful people. They couldn't have been better hosts to me. So this photo is the two of us in her grandmother's house, which is in a small village near Kuala Lipis. And there, you can just about make out, I'm sorry, it's a very blurry photo. That is us on a rakit, traditional bamboo raft, crossing the river Pahang. Okay, I think this is something everybody should do at least once. It's a lot of fun. You know, all the beetles and stuff are running up your legs, but you can't move because the thing might capsize. And so it was interesting. And also, the reason I've got the little arrow there is to remind me to say that I was later told that there's this saying that if you have been in the river Pahang, then you must come back. And so I think that has certainly turned out to be true for me. During my undergraduate training, I took the opportunity to do a 12-month industrial placement. And this was something that Surrey offered. And this was where Yasmin and my paths separated because it didn't make sense for her to do this training. She continued and graduated a year ahead of me. I spent a year working at what was then Beecham Pharmaceuticals. It's now part of SmithKline Beecham Group, in where their main research was in Brockham Park in Surrey. That, I think, has since closed down. But Brockham Park was a wonderful place to work. They had amazing research labs, not just this old house that you see at the front. And historically, it was very interesting because it was the place where the structure of one of those antibiotics I mentioned earlier, penicillin, probably the most important and best-known antibiotic, was discovered. And my job when I was there was working with a group that were testing new formulations of penicillin against various microbes. This was a pivotal year for me because I loved research. I really discovered that was what I wanted to do. And it also made me decide that I had to do a PhD, because I could see that if I wanted to drive my own research, if I wanted to be a group leader, that's what I'd have to do. So after I returned to university and graduated, and this is a picture with some of my friends from my graduating year. Actually, I'm the only one from microbiology. Anne in the middle and Belinda were biochemists. Is Dr. Nolida here? They're classmates of Dr. Nolida, who was another one of our ISB lecturers who was in Surrey, and Colin, who was an engineering student. So I applied for scholarships and won one from Whitbread to go to Cranfield University to do my PhD. And actually, to sort of explain this a little bit better, Cranfield University had a very strong biotechnology program, but their strength was mostly in the bioprocess engineering end. But they were collaborating with another group of biological scientists at the University of Leicester in a place called the Leicester Biocentre. And Leicester was where all the gene cloning stuff was hotting up. And so for those students like myself, there was a group of us who were doing more gene cloning. They seconded us to Leicester University. So I was actually based at the University of Leicester for my PhD. And there I was working on industrial yeast. And so here's a picture of some yeast, microbe. And this is just one photograph from my PhD. So this is an agar plate, and these wiggly lines are where the yeast cells are growing. And in this particular plate, we put starch. And then after the yeast have been allowed to grow, we expose the plate to iodine vapor. And so where there is starch present, it will turn black. And where starch has been degraded, you get this zone of clearing. So this is the industrial yeast, and this is the industrial yeast after we've genetically engineered it. And what we did here was we added a gene from a plant, wheat alpha amylase, that can break down starch to make sugars, and then the yeast can now metabolize sugars and be much more efficient. So it was part of the work I did in my PhD. This was not my paper. This was the group that actually cloned that gene from wheat, David Balcomb's group. And I mention his name now because I'll come back to him later on, and just another small world connection. I still collaborate with University of Leicester, but not on yeast, now on plants with Pat Heslop-Harrison. But when I go back to visit him, I also catch up with some of my old lab mates. There's a few still kicking around Leicester. My PhD supervisor, who's now retired, Chris Hadfield, Pat and Ian Loft, who were in the lab with me when I was doing my PhD, Peter Meacock here with his wife Jean, and Peter Meacock was the leader of the yeast group at the time I did my PhD. And I also met somebody else in Leicester, another Malaysian. And this one, I've named him as maybe a major catalyst. As biologists, we have to use these technical terms, turning the English kampung girl into the Malaysian kampung girl, or if you will, the English bride into a Malaysian bride. And probably a major incentive for me to end up in Malaysia. But we didn't go straight to Malaysia. We decided that we would like to do some postdoctoral research. And many of the people in Leicester, the postdocs there, had been working in the United States, and they were telling us how amazing it was to work there, so we applied for positions. And I got a postdoctoral fellowship at University of California, San Francisco, so here we are trying to look Californian cool in our sunglasses. I was in the lab of a very brilliant pediatric endocrinologist, Walter Miller, and he was working on human systems and enzymes involved in cholesterol and cholesterol metabolism. And he hired me because he wanted somebody who was used to cloning genes using yeast, because he wanted to use yeast as a model system for some enzyme engineering studies. And those went pretty well. We got some patents and some very nice papers out of that work. Harry wasn't he was in California with me. He wasn't in San Francisco with me. He was doing his postdoc about 80 miles away in University of California, Davis, but we used to meet up on the weekends. And it was in Davis we were to meet another Malaysian, and here is Professor Helen Nair, and she was over there doing her sabbatical. And so then through that friendship with Helen, when we did eventually move to Malaysia, it was to Helen I reached out, and probably another reason that I ended up in University of Malaya. So I worked with Helen for a little while, and then I joined Dato Saleh's new company, Trop Bio Research. And as Prof. Yasmin has mentioned, he really was a pioneer. This was the first plant biotechnology company in Malaysia. And there were a whole bunch of fun people to work with there. I worked there for about 5 years. You might recognize some of the people are not known. They were in TropBio before. Many people may know Dr. Kodaswaran, who is now with Bioeconomy Corp. Anybody recognize this guy? I think he's not able to be here because he's very important in the audit which is going on now. Professor Fong Manyik. Oh, he's here. Thank you. Oh, I'm so happy you're here. Okay. And Chu Fun. Did Chu Fun make it? She's now in University of Nottingham in Malaysia. We worked on many different projects there, but the ones that I particularly remember were some of the DANSED projects, Danish Corporation for Environmental Development. And perhaps my favorite of that was working with Jens and Mona. And I'm still in touch with them on Facebook, so if they happen to watch the video, hi. But as Yasmin mentioned, I really had a strong calling to be in academia. And it was through Tan Sri Syed Jalaluddin, who was then the Vice Chancellor of UPM, that I had the opportunity to get an academic post. So my first academic post in Malaysia, which was as an associate professor at UPM. In UPM, I started writing grants furiously and trying to get my work going on abiotic stress, which, as I mentioned, was the topic I was very interested in. And this was work I did together with Professor Ho Chai Ling. I know she's here today. And also, this was work done by our graduate students, Dang, who was a PhD student from Vietnam, and Yin Yi, who was a local master student. And what we were doing here is we actually chose to look at plants that live in the mangrove. These are incredibly hardy. They can live in salt water and really bad nutrient systems, and yet still thrive. So we wanted to look at the genes in these plants and see if they might be useful for agriculture. When I was in UPM, Harry, as he has to do, went overseas again, and he got a sabbatical at MIT, and he was in Tony Sinsky's lab for about 6 months. Professor Sinsky, his wife, Professor Rar, is another professor at MIT. And when she found out from Harry that his wife was also an academic, she said, well, you must invite her over. She has to come and work in my lab. So she kindly invited me, and UPM and the program that Yasmin mentioned permitted for me to go there for a 3-month fellowship. Here I did some work, again, using yeast, back to working on yeast. And it was, this is actually my very first microarray. So for those of you not familiar with genomes, we actually have lots and lots of genes in a cell. And the microarray was the first technology where you could put every gene of an organism on a single chip. And to put it in context, this is about the size of a 5-cent coin. It's a very small area. And in yeast, there's around 6,000 genes fit into that little space. And we treated yeast with tocotrienols, which are extracted from palm oil. They're very powerful, very good antioxidants. Take them, they're good for you. And we wanted to see then how this changed the expression of genes. And since we knew what all the genes are in yeast, this was one of the model systems we could use. When I was in MIT, I had the opportunity to attend classes and meet students from MIT. And I was really impressed with their teaching models and methods. So when I heard that there was a new university being established in Malaysia that was going to teach curricula from MIT, I wanted to join and did, and that was the Malaysia University of Science and Technology. In MUST, we only had postgraduate students. They were all master's students by taught course, but they all did some research as well. And because we had good funding initially, we were able to offer scholarships and attract some really bright students. And many of these students now are actually academics in Malaysia, and I'm not sure if many of them are here. Examples I can think of are Lionel In, who is now a lecturer in UCSI, Adeline Song, Lionel and Adnid, there they are, wave to them, academic in UPM. Also Xiao Hun, who is a lecturer in Utah. And 3 of these students actually did PhDs with me. Wai Ting, who is now doing her postdoc in Taiwan. Hui Li, who has just submitted her thesis, and Kai Swan, who I hope will submit his thesis very soon. MUST is just down the road in Klanajaya. So this was when I managed to finally get back in touch with Yasmin again in UM. And so we started working together. And this work was work that Yasmin was doing to use plants as systems to express proteins. And some of the proteins she was interested in looking at were antibodies. Now, plants are not mammals. They don't have antibodies. But if we put the mammalian genes to make antibodies into the plants, we can use them as a production system. And these particular projects here were actually making antibodies that recognize plant viruses to use them as a novel way to protect plants against viruses. And this work was the PhD work of Chua Kek Heng, who is already a professor in University of Malaya. Okay. And this was also when I had my first introduction to Professor Nosalani, who is now a good colleague and friend. And the supervisor on the MUST side was So Hun, who is now in Melbourne. And the other students doing the work were Diana and Adria. Actually, Diana was an MUST student. She's now in Singapore. And Adria was a technician for me under that project and then joined me and did her master's later on in UM. So while I was in MUST, my interactions and connections with UM kept getting stronger and stronger, and I was actually started to do some part-time teaching because some of the senior lecturers had retired. And eventually in 2006, I moved on a full-time basis into UM and started my work on microRNA. So now I've taken you around the tour, and from here on, I'm in UM, 2006. So back at UM, balancing act. This slide will be familiar to any academic. How do they expect us to do everything? And I also put this kind of gear diagram here because we have to make everything fit. We have to make everything work together. And it's to remind me that in 2006, UM was gearing up. We were really cranking up on research at that time. But it was good because if you wanted to do research, there was quite a lot of research funding available. So you wrote the grants, you wrote good proposals, you could get money to do research. And so all of us did. So I was a lecturer in Institute of Biological Sciences, and I still teach in Institute of Biological Sciences today. I teach microbial genetics and microbial processes. And here's just one of many batches of students, but many of this batch, I think this is Sue E's batch, who's my research officer. Is that your batch? Yeah, I think you're in there. And I think we've got Suba and Ching Ching, who's the current coordinator, and Lani and me and Yasmin here, and Hashima, all on the front row. And as Yasmin mentioned, I was a coordinator of that program, and here's some of the lecturers sitting around the table. Now, during that 2 years from 2007 to 2009, that was the time that the program was undergoing the curriculum formatting into MQF. I think some of the academics may remember it was a painful learning experience. Doubly so for me, because if you think about it, not only had to be done in English, it had to be done in Bahasa Malaysia as well. And I kept my colleagues in stitches with my terrible translations. Google Translate didn't used to be so good in the old days. They'd be rolling around. At least it brought some levity into the meetings. And they say, no, Jenny, that's just not right. Okay. And thanks to them, really thanks to them, I had an awful lot of help. And especially, I think here I want to mention Hashima. Hashima had the misfortune to have the office next to mine. So I was always going and knocking on the door. Hashima, what do I do with this? And she never turned me away. She's the patience of a saint. And so we managed somehow to get that program together. And we continue to upgrade and work on the curriculum. So that's actually now quite enjoyable. So I learned a lot about teaching from going through that experience. I'd started to work together with a bunch of people who were in this Center for Research in Biotechnology for Agriculture, or SEBA as we call it. And Yasmin was actually the initial head of that group. And then after a while, Nozolani took over as head. And then Nozolani was promoted to being one of the cluster deans, and Yasmin and Lani decided that I should be the head. So I could say initially I was quite worried about taking this on. But they promised and delivered on their promise to give me tremendous support. And in fact, all the people in Sabah, many of the people in Sabah are actually also in the Institute of Science, Biological Sciences, are incredibly supportive. We also have some wonderful support staff shown here. And as of last year, we actually have new lecturers like Dr. Katharina, Dr. Bunchin, and Dr. Chihao. Chihao, you're on a later photograph, not this one. 2009, when I took over as the head, was a very important year for Sabah because that was the year we launched our transgenic greenhouse. This was the first such facility in Malaysia, and it was the first to get international certification for compliance to operational and containment standards. And we use the Australia and New Zealand standards because till now Malaysia still doesn't have standards. And it has been continuously certified since then and remains the only fully certified and continuously certified facility in the country. And we're very proud of that. It takes a lot of work, and I have a wonderful team helping me to do that. So when you're doing all of that, how do you get your research done? You have to get people to help. You have to work with others. You have to collaborate. And this is a message I will say over and over again. If you're open to collaboration, everything is possible. And so how do you know who to collaborate with? Well, luckily it's a small world. In Malaysia, I think all the plant scientists know each other, and we meet at meetings and we talk and we say, how about working together on this or that? So some of the people I have been and still am working with, Professor Wiki from UKM. We work together on acacia and on rice. And one of her students, BK Song, who is now a lecturer in Monash University, still collaborates with me and with Professor Sadek. I can see him sitting back there. And we're co-supervising student Mohana, PhD student, working now on microRNAs in rice. Also collaborators from Mardi, like Dr. Tan Chon Sen. And one of the highlights of that time working together with Vicky and with Academy of Sciences Malaysia, they kindly helped us to bring over Prof. Gudev Kush, who for anyone in the field of rice will really appreciate, really the father of rice genetics and rice breeding. And he was very kind to come over and do a workshop with us. And also Chai Ling is here, and next to me, Umi, who was then in Mardi, but I think now is with Crops for the Future. On microbiology collaboration, I've worked very much with Professor Yo Chu Cheng. He was my colleague in MUST and is now a professor in UNISA. And before he joined MUST, he was in NUS. As he hooked him is here from NUS, so a little wave to NUS. And he had pioneered work on toxins and antitoxins in bacterial systems. And so he was working on characterizing this toxin and antitoxin in a human pathogen, Streptococcus pneumoniae. And this was work by a PhD student that we co-supervised. So she's a PhD student from University of Malaya, Whiting. And after this, we decided to see, well, I work on plants. Can these systems work in plants? So we tested whether these bacterial toxins and antitoxins would actually function in plants. And this is the work of PhD student Fauzia Abu Bakar. Fauzia, there at the back. Hi, Fauzia. Fauzia is now a lecturer at Quest International University in Perak. And she did some really beautiful work showing that the toxin actually does work in the plants, causing cell death, and that the antitoxin can neutralize that toxin in plants. And we also went on to show that that works in algae, and this was the work of another student, master student Shet Li. Shet Li is just won a MEC scholarship and is in Japan in Nara Institute of Science and Technology doing her PhD now. And of course, I worked with people in University of Malaya as well. And this, I mentioned Hashima, my colleague next door, and my colleague one block away, Suba. Now, I was very excited about microRNAs, so I was telling everybody, you've got to work on these, whatever you're doing. I'm going to do them in plants. You've got to work on them. Then I persuaded Hashima and Suba also to get excited about microRNAs. So here's a paper related to microRNA in cancers, and this was the work of Nora Hayu. That was her master's project. She's just finished her PhD and graduated this year. And in giant freshwater prawns, which are delicious, by the way. This is Suba's work, and the student doing that work for a master's project was Chan Chan. Chan Chan has finished her PhD at Nara and is now back in Sabah as a postdoc. So I mentioned NICE just now. It's one of many places around the world that we have collaborations with. I'm not going to mention all of these, but I'm going to pick a few. Together with Yasmin and Tio, we're working on a Bill and Melinda Gates Foundation program under IITA. And Ivan is over there, a PhD student from Uganda, working on East African highland bananas. I continue to work with Pat Heslop-Harrison and Trudy Schwarzacker from University of Leicester. We've started collaboration with ICGEB New Delhi in India. Is Jazz here? Yes, Jazz is about to go there next week for a 6-month fellowship with Dr. Neeti. And we're working with various universities, Mike Jones in Murdoch University, on biosecurity in crops. So I hope I've convinced you collaboration is important, and we have lots of it going on. And with other people in Sabah, we have many, many more collaborations. Lately, I've noticed that you keep hearing this thing in radio adverts and TV adverts. It's in our DNA. Everybody knows it. They don't know. They might be talking about a car or all sorts of things, but everybody now knows about DNA. And this is great for biologists, because we know that when we say DNA, click, people know what we're talking about. It's in our DNA. And in a way, everything is in our DNA. It's the double helix, everybody knows, in your cell that has the information that's going to tell your cell whether it belongs in a plant, this is a banana, or a microbe like yeast, or a person like us. DNA is actually where all the information is stored. So yes, it is in our DNA, but in order to get from here to actual molecules like this, proteins that are the enzymes that actually do all the work in the cells, you have to go through this middleman here, the RNA, this single-stranded guy. So I would say it's in our RNA. Okay, we've got everybody saying it's in our DNA. The next job will be to get them to understand it's in our RNA, and then the biologists can take over the world. So if the DNA has all the information a cell will ever need, then the RNA has all the information that cell needs right now. Okay, so if we want to know what's going on in biology, this is what we have to look at, what's happening in the cell now. And we call this set of RNAs the transcriptome, because this process of changing the DNA into RNA is called transcription. So transcriptome is this full set of RNA molecules. And they're going to give us a really quite good idea of what is going on in the cell, which proteins it's likely to make, and then from that we can extrapolate to other things. So the transcriptome tells us what's happening now. It's really important to study it. But we need to actually not just have the sequences, we actually need to know how many there are and what the sequences are, and that turns out to be quite complicated to do. So you've probably heard of genome. This is describing the full set of the DNA, and the transcriptome is the full set of RNA in particular cells at particular times or particular conditions. So in our group, and I have to say it's not just me, it takes a lot of people to do all this work, we try to look at genomes and transcriptomes, where the motivation is to study the messenger RNA populations and know all the nucleic acid sequences, the genome sequences, so that we can go on and do the functional studies. So objectives of this kind of work is to identify and annotate genomes and transcriptomes. And the plants we were looking at for this banana, oil palm, and fingerroot ginger. Okay, so this is some work from PhD students Ranga and Wan Sin, master student Ron, and below here the oil palm work is with Ki Bin. So here we were looking at bananas on the top here, these two papers. Now, bananas mostly have, most of the bananas most people are familiar with have a type of genome called the A genome, and they have 3 copies of it, so they're called AAA. And this would be your Barangan, your Cavendish type banana. There are other bananas, like the cooking bananas, that we have the B genome. And some of the bananas we eat in Malaysia have a combination. So your Rastali is an AAB, for example. So they have slightly different sets of genes and sequences. So we've actually worked on both the bee genome. This was work we did together with Mark Davey in KU Leuven. He's now moved to Bayer Crop Science. And we looked at all the microRNAs in the A genome. We didn't do the genome sequence. Pat Heslop-Harrison, one of our collaborators, was involved in that project, which was a very big genome project. And Ki Bin, on the other hand, is looking at the oil palm genome, and he's a Sime Darby staff as well as a University of Malaya PhD student. And what he's doing is trying to see all these different markers in the genome and how they can be used for plant improvement. So some of the main findings from this work was the identification and annotation, kind of giving functional labels to all of the microRNAs in the A and B genomes of banana, and demonstrations of genome-wide markers usage in breeding in oil palm. The next project I'm going to talk about was an HIR project, and we are in the HIR building here today. And this is on a plant called Bosenbergia rotunda. Now, I cannot claim to have been the one to start working on Bosenbergia rotunda. There's a very strong group in University of Malaya with interest in this plant. In Malay, it's known as Temu Kunci, or fingerroot ginger, and this is a picture of the rhizome, which is the part people use in medicine or even in cooking. We had HIR icon, Pat Heslop-Harrison, and his wife, another collaborator, Trudy Shawsaka. PhD students, Sherming, who was actually, Lani was the main supervisor. Rezo, who's just graduated on time, I have to add. Theo is helping us on this project. Tyson is going to submit his PhD next year. Teresa has finished her master's. She's here somewhere doing metabolomics. Now the Sime Darby staff here. So in this project, which is a very big and ambitious project, and we haven't actually finished, we're still working on it, but some of the things we've done, we've described several transcriptomes from several different kinds of tissues, methylome, which is to do with the methylation status of the DNA, metabolome, we've characterized SSR markers, and we are working on the genome assembly. The reason it isn't finished is when we started working on this plant, nobody had actually looked at the genome, and we thought it wouldn't be very big. It turns out to be 5 times bigger than we thought it was. So that actually costs like 5 times as much to sequence, so it's taking us a long time to find the money to finish it up. So this a little bit about genomic and transcriptomic studies. I'm now going to start finally talking about the small RNAs. And one of the things that small RNAs are very important for is a phenomenon known as RNA silencing. So far I told you that RNA was important to be used to make proteins, which is true, but that's not all it does. It's actually involved in many other things. It's actually involved in several parts of the protein-making process. But the RNAs I'm interested in that are involved in silencing are actually involved in the regulation of the use of RNA. And this phenomenon of actually stopping the RNA being made into protein is what we call gene silencing or RNA silencing. So probably the first observations of RNA silencing, and this is taken from a review by David Bolcom that he wrote in 2004, he points to this paper from 1928 by a guy called Wingard. And Wingard was working on viruses in plants. And this is a tobacco plant infected with a tobacco virus. And what you can see is the first leaves that have emerged when the plant was infected, showing lots of symptoms. But as the plant grew and matured, the new leaves that are coming out don't have symptoms. It's as if the plant has been cured. But what Wingard also found was if you try to infect these leaves, you can't infect them. They just don't get infected. They're protected somehow. And we now know that that was due to RNA, but at the time, it was not known. It took a very long time to actually figure out what this mechanism was. And most famously, the work of Craig Fire and Andrew Mello in their paper in Nature in 1998. They were working in C. elegans, a worm. They actually showed that it was double-stranded RNA that is a trigger to do this RNA silencing. So if you remember, messenger RNA is actually a single strand. When you have a double strand, that's unusual, or it's seen as something different. It's not seen as something to make protein, it's seen as something to trigger this RNA silencing. And it's cut into smaller pieces by other enzymes. And then based on the nucleic acid sequences matching this single strand of the double strand together with the single strand of the messenger RNA in a sequence-specific manner, it will cause this gene silencing. So it's a very selective and precise process. So Fire and Mello won the Nobel Prize in 2006 for their paper in 1998. What the plant scientists will very happily or annoyingly point out to you, there's this paper in 1997, Radcliffe et al., which was David Bolcom's research group 1 year earlier in Science journal. They had shown that the trigger for RNA silencing was a cytoplasmic double-stranded RNA, and not only that it silenced the genes inside the plant, it was also what was silencing the virus. It explained what was going on in Wingard's plants back in 1928. But they showed it in plants, and there's no Nobel Prize for plants. Okay. But don't give up or stop working on plants, because there's a happy ending to this story. Okay. Because they published the mechanism first in 1997, and because it is the identical mechanism, they have the patent. So any money made out of this goes to these guys. Not only that, in recognition of his work and many other good work that he's done, David Bolcom was knighted by Her Majesty Queen Elizabeth II. It's now Sir David Bolcom. So who needs a Nobel Prize if you're a knight? I mean, it's a bit better, I think. So these triggers for silencing are double-stranded RNAs. They can come from viruses, and they can come from these, finally, you get to see them, microRNAs. Now, microRNAs are actually made inside all multicellular organisms, and they are actually transcribed as a single-stranded RNA, but they're able to fold themselves so they look like a double-stranded RNA, or in fact, that part of the RNA is double-stranded. And then these can trigger this RNA silencing, where they will match to a messenger RNA with a similar sequence and stop it from being made into a protein. This silencing is sequence specific. So that's really important, because that means if we can find all of these microRNA sequences and we know what other sequence then they are going to silence, so we can predict which genes they're going to regulate. So this is wonderful for anyone with a computer and a bit of bioinformatic knowledge. We can take all these sequences, find all the matching sequences, and we know what's going to go on. So we started to do some studies with this, and I'm going to tell you about 3 sets of studies. The first in oil palm, which is the largest crop and arguably the most important crop in Malaysia, and bananas, which is the most widely eaten fruit on the planet and a very popular fruit in Malaysia, and orchids, well, because they're pretty. So our work on small RNAs in oil palm has focused on flower development. And I probably don't have to justify, especially with Tan Sri Augustine Ong sitting in front of me, who's a great proponent for oil palm. I don't have to justify why oil palm is this amazing crop. It is economically the biggest crop in land and value. And despite all the bad press it gets, it's actually a really ecologically good crop. If we are going to have vegetable oil, and I can't find anybody who could argue that we can live without vegetable oil, if you were going to produce it from canola, you would have to use 3 times as much land. If you were going to produce it from sunflower, you would use 6 times as much land. And if you were going to use soybean, it's 8 times as much land. How is oil palm not more sustainable? I don't know. And it's also a very rich biodiversity. If you walk through a field of sunflowers or soybeans and try and catch animals, I don't think you'll find all of these fellas. Okay, this is what you can find. Some of what you can find in an oil palm plantation. Now, I lived in an oil palm, thanks to my wonderful husband, in an oil palm plantation for 4 years. And at some time or other, we've had all of these critters in the house. Have you ever had a monitor lizard in your bath? Yes, I can't recommend it. And I really don't like spiders, and they get pretty big out there in the plantation. So, and one night we had one of these. We got woken up, Harry will remember this, we were woken up by frantic barking from our dogs, and he went out with a torchlight to find this scaly, hissing ball and was terrified that it was a snake. It was, in fact, a pangolin that had rolled up, and Harry valiantly rescued it and released it unharmed into the plantation. And we've even had binturangs in the garden, beautiful, beautiful creatures. That's an aside. What I'm actually interested in oil palm, though, is working on the flowers. And why we work on the flowers is because since the product is the oil, and the oil comes from the fruit, then the flowers, and in particular the female flowers, are very important. So the motivation for this research is there's a need to improve oil palm even further. We might say it's a sustainable crop, but we still have more people and less land, so we have to become increasingly efficient in our production. We can't stop even saying it's a good crop. And so the research questions that we were looking at was what are the small RNAs in oil palm, and in particular which ones are related to flower development. I just want to give you an indication of how hard it is to work on these kind of plant systems, especially for somebody who was formerly a microbiologist. You know, microbiologists stick a few cells in some broth, come back, your experiment's finished in a day. Oil palm, not so fast. Okay. So if we want to look at the flowers, here we are actually sacrificing a tree, or sorry, I should say a palm. They're not a tree, apparently, botanically. We have to chop up this stem and extract from inside of each leaf the part that will eventually develop into the flower. And if we label the frond that we can see as the youngest frond as number 1, we go upwards 2, 3, 4, 5 with the older, and we go downwards inside the stem, and we went back all the way to minus 27. And at this point, it's pretty small. We have absolutely no idea whether that will be a male or a female flower. So in oil palm, you have both male and female flowers on the same palm. So we have to quickly put that into liquid nitrogen. This was my student Saha here. Bring the lab out to the field, get them into liquid nitrogen, and then bring them back to the lab and look at the RNAs. So this work was done by Nadira, my master student, who's the staff of Saimdavi. Saha was a master student from Iran, is now in Canada. Hui Li, my PhD student, who's just submitted her thesis, and Joel, another PhD student, and also Saimdavi staff. So our main findings, we've identified many microRNAs from oil palm, and we've classified the ones that are associated with flower development. And apart from the microRNAs, we've classified transcription factors that are associated with the male flowers and the female flowers, and just generally with flowering. Our work on banana, I've gone back to abiotic stress again. I think everybody loves banana. It's a favorite fruit around the world. But in the countries that grow it, mostly it's not exported. About 80 to 90% is actually a staple food. So it's not just important as a dessert banana. It's actually an important main part of many people's diets. And it's quite susceptible to flooding and salt water. So we wanted to look at whether we could identify genes involved in that reaction to salinity. So we need this knowledge if we're going to mitigate abiotic stress in crops like banana. And the research questions here were which are the small RNAs in banana and which ones are related to stress responses. So it's another big group of people. Most of the work is done by 2 PhD students, Ranga and Wansin. Wansin has graduated. Ranga is working on his thesis now. Here again we have Ron, who's finishing up his master's on characterizing some of these. Some of my postdocs, Purabi and Pooja, research officer, Sui, and various, Jazz is here, and some interns that we had in the lab. And this is also very much work with Professor Yasmin, Professor Nozolani, and Dr. Teo, and Ivan, as I've already mentioned, working on the East African highland bananas. So some of the work we've reported more recently, we've annotated the microRNAs in about 181 of them. 56 of them are new in banana, and around 60 of them are related to salinity. And from knowing which genes they're regulating, we can see which pathways they're affecting, and this is related to stress signaling and defense, transport, cell homeostasis, and metabolism. The work we're doing now is we're taking some of these genes and we're overexpressing them in model plant systems. And we've got Ron has some really nice data. I'm just looking at of that actually causing some drought tolerance in the model plant system. So we're hoping to publish that very soon. The very last part of my talk, I would like to tell you a little bit about using RNA silencing as a tool for biotechnology. So when we have a double-stranded RNA, we can find out what it's silencing. We can also go backwards. If we want to silence the sequence, we can make a double-stranded RNA that matches it to deliberately silence the sequence. So we can design these double-stranded RNAs and use them in biotech. And this is some work we did in orchids. Orchids are beautiful. They grow very slowly. And if we were to do genetic modification by putting new genes into them, if I was to do that today, I probably have to wait maybe 4 or 5 years to know what the flowers look like. It's just way too slow, and it makes it a very difficult system. And if we do that also, we would then have a genetically modified plant, and there's certain regulatory hurdles that still remain for use of these, even in ornamental crops. So our motivation for this part of the research was we know double-stranded RNA will silence genes, but can we silence genes by directly putting the double-stranded RNA on the plant? Not by making the plant make double-stranded RNA, but we make the double-stranded RNA in bacteria and put that straight on the plant and see if we can cause a silencing. The first study, we decided to target a virus, Cymbidium mosaic virus. When viruses infect you or plant cells, they're evil things. They take over your whole system and turn you into a factory to make more virus particles. So in the case of Cymbidium mosaic virus, it does that by making the cells make more of the coat protein, and then it uses the coat protein to make more virus particles. And as these particles are made, that's why you get these symptoms. So you can see this would totally destroy the flower. It has no economic value once it's infected. So our model here was we will take double-stranded RNA that matches that coat protein messenger RNA, and we will put it onto our plants. This should trigger silencing of the messenger RNA for the coat protein and stop the coat protein being made. If there's no coat protein made, then there's no virus, and if there's no virus, the plant should be healthy. So here I'm just showing you some data. The bars indicate the amount of RNA of the coat protein that we measure using an assay called quantitative PCR. And we have 6 different groups of plants. So I start by looking at groups 5 and 6. 5 and 6 were not infected with virus at all. And so the levels of the coat protein, since they don't have virus, they shouldn't have any coat protein. So this sets your baseline. This level means no coat protein. On the other end, we have the cells that we infected, or the plants we infected with virus, and we didn't give them any RNA treatment. And so this is the level of coat protein you have in infected samples. And in the middle, we have the samples that were treated with single-stranded sense, single-stranded antisense, or double-stranded RNA. And I hope you can see that when we use double-stranded RNA, we have effectively no coat protein RNA, which means effectively no virus in the plants. So these are some of the medals Prof has been mentioned, and she's actually my co-medalist, so she's allowed to tell you about those. And we've patented this. Our patent was granted in 2015, and we've just registered the trademark as well for this. So if putting double-stranded RNA straight onto the plant can protect against viruses, that means it is getting into the cells because all that viral replication is occurring inside the plant cells. So we should be able to use that same method to silence the plant's own genes. So can we use this technology to modify orchid flowers? So the first thing we have to do is we have to have a gene to target. And so we looked at, when I say we, I should say Sui, this was her master's project, looked at genes that are being used as the flower goes through its different stages of development. And also Trudy Schwartz-Zacho helped us a lot on the botanical parts of this study. And then we, having decided which gene is involved in flowering, we make double-stranded RNA and treat the buds at an early stage. Hopefully this silencing goes on, and then we look at the flowers we get at the end to see what has happened. So we only had a small grant, so we only did one gene. It's called MIB1. It's from the MIB family of transcription factors. And it's also known in other plants to be associated with flowering. And we find that, again, this is the same assay. We're looking at the amount of RNA at different stages of flowering. And we could see that this is a gene that's definitely associated with flowering. So when we treat the orchid buds with double-stranded RNA against that gene, and this is the bud on the blue bar, in each case, if we don't treat, that's the level of the RNA. And when we do treat, we definitely reduced the amount of this RNA. But what happens to the flowers? Well, Sui spent many days in the greenhouse and always coming back to me and saying, I think I might see something. Maybe there's a slight difference. But we really couldn't see anything convincing. And then I said to her, maybe we're not looking close enough. So this is another story where small comes in. You have to look for the small changes. And we had to look really small. We had to use electron microscopy and really go down to looking at the petals and the shapes of the cells on the petals. And what we found was that in this part of the flower, the labellum or the lip of the orchid, when the cells are normal, not treated with any RNA, they are quite lumpy and bumpy or sort of conical shape. And when they had been treated with the RNA, they became very flat. So we definitely are able to change the shape of the cells, and we are able to have an effect, even though we can't see any real difference at the level of the flower with that particular gene. And so this is the work also of Adeline. She was involved in part of that study, and Trudy in Leicester University. So what we found from this study is direct use of RNA. It can protect orchids against viruses, and it can result in changes in endogenous genes that can change cells. So it has a good potential for use for genetic modification without waiting to genetically engineer the plants. So it'll be faster and less expensive, and we don't have to worry about the plants being transgenic. Okay, I got to my closing remarks slide. A little bit more to go, folks. Okay. So I try to, I think Yasmin will give a longer summary perhaps, but I'll just try to give a few important points that I feel. Renewable resources are really important. It is just this one planet we're all living on. And a big part of that is using everything efficiently, sustainably. And I think we need more people to work on that. One of my bugs when we talk to final year students is, I want to cure cancer. I mean, it's good. We do need to cure cancer. But everybody has to eat every day. You know, isn't that a bigger problem? Okay. Collaboration is really key. I mean, all of this work I've been able to do, there's no way I could do it without a big cast, a big supporting cast. Colleagues, students, postdocs, and friends overseas, conversations at meetings where we get our ideas. So it's really, really important to work with others. And anyway, it's more fun working as part of a team. Silencing is important. It's golden. You know, a little bit of a change in a small piece of RNA can actually have quite a big effect on a phenotype, on how things appear. And so it is interesting and worth looking at. And this means that every small thing matters. Sometimes when we're doing research, we feel like, oh, what I'm doing, you know, I'm not curing cancer, I'm not saving the world. But every little thing we do is important. So just do whatever you can do as well as you can do it, and you can make a difference. And for this, I like to use the Malay proverb, sikit-sikit, lama-lama, jadi bukit. So if each of us does a little bit, it can mean a lot. I have a last few notes for you, and they're going to be musical notes, if I dare. One of the wonderful things, or among wonderful things in University of Malaya is incredible things we have around us. We have this beautiful campus, we have botanical gardens, we have museums, we have art galleries, and did you know we have a symphony orchestra? Of course, yes. And run under the Cultural Center by the very talented Dr. Nazir. Thank you for coming today. So it's really been a joy for me to have the opportunity to get back to music again and play with the University Orchestra, which is really a lot of fun. I enjoy it a lot. So today I have the stage to myself, and if I may, I'd like to play a short solo for you. Syrinx is a mythological character. Pan is fascinated by her and he chases her through the forest, but he loses her when she hides in a bed of reeds. And frustrated that he can't find her, he takes some of the reeds and makes a flute. I like to think of this as a kind of metaphor for scientific research. We're often chasing something that is deeply fascinating, but rather elusive. Okay, I'm going to use music because I'm a bit nervous. So this is Syrinx by Debussy. Sorry, too sweaty. And I have to thank many more people that are on this slide. I tried to get in as many as I could. And one person, or a few people, I have to have special mention, of course, my husband, my wonderful mum-in-law, Neela, and Jelly, my treasure, my home manager, without whom I don't think I could live. She's helped me bring up my kids and manage my house. So thank you.
Thank you, Professor Jennifer, for sharing with us such a wonderful journey of your life in pictures and in music and in research. Ladies and gentlemen, I would like to call Professor Rofina to summarize and conclude today's inaugural lecture. Please welcome.
Thank you very much. So such is the talent of Professor Jennifer and Harikrishna. Join me before I start again in congratulating her on such a fascinating and insightful talk. It's a small world after all, Jennifer. Definitely, we've seen how she has grown from a small kampong girl in Nottingham to her new kampong, and how glad we are that she has joined us in our kampong here in University of Malaya. And we know from what she has told us that an academic career is not built in big leaps, but in small steps, and involves so many others along the way. A researcher is enabled by encounters, chance encounters, small encounters at meetings, at events, and connections that they will develop into collaborations and lifelong friendships. And as research is so wide and deep, over time, one may end as an expert in some topic, but there are small groups of people who again and again will create the small links that will really make the connections that impact us again and again, and hence the small world that we live in. She has shared with us today her work on microbes, on microarray, and very significantly, and she downplays this a lot, her significant contribution to the work of small RNA in plants. And I think we should all congratulate her on her wonderful achievements in this area. Her career journey from the start of her PhD, her award-winning PhD, throughout her years as a postdoctoral researcher, to her work as an academic in UPM, in TropBio, in MUST, and eventually in the University of Malaya, really highlights how she has developed as a scientist and how she has contributed in bringing us all together in this pursuit of knowledge. And what a haunting melody you played us, Professor Jennifer. And I really agree with her. This is sometimes, a search for the unknown, but it's passion that drives her. And you can see very well how a passionate scientist carries on and carries on and carries on with the support and collaboration of all those around her. So a central theme of what Prof. Jennifer has brought us today is that collaboration is really key. And really her ability to get everyone around her excited by what she does is one of the key attributes to Professor Jennifer. In her leadership in SEBA, as we have seen, she has really allowed the center to grow beyond what it has originally been created for. And her achievement in defining SEBA as one of the leading centers for biosafety actually shouldn't be underplayed as well. SEBA now is collaborating with international agencies and international universities for biosafety and biosecurity. Many of our members now sit on regulatory committees and boards at the national, international level. And we now are trying to drive also policy decisions through the work that she has done with the biosafety team in SEBA. Onwards to what she has done in science. As I said before, she has managed to get everyone excited about these small molecules. All of us are now contaminated by this desire to work with small RNAs. And all of us have pivoted our science to this very exciting world. And really, the discovery of small RNA, of course, as she mentioned, given the Nobel Prize to the people in medical research doing cancer, belies the fact that the whole discovery was done in plants, and plants remain at the forefront of discovery. We discover so much more in plant systems than in animal or human systems that now have been translated to use in both mammalian and human research. And so, so for the young scientists, I hope you can see the impact that plant science has made on our advancement of knowledge. And Prof. Jennifer remains at the forefront of introducing small RNA research in Malaysia, and even I could say in Southeast Asia, if you look at the depth of her papers and the depth of her publications in this field. Her work on oil palm, for instance, has identified regulatory mRNA associated with a very important aspect of our economy, which is on the flowering of oil palm, the ability for palms to produce fruits faster. Her work on abiotic stress in bananas. Bananas is our favorite fruit. And don't forget, this is the center of origin of bananas, is Malaysia, and we need to be in the forefront of the research as well. And her work on abiotic stress in bananas, in improving banana cultivar, has really taken us to the next stage. On RNA space in orchids, those beautiful plants, as she said, so these are on the cusp of being commercialized and having an impact on industry. And her work on small RNA continues again to go and to impact all of us in this university and also in our community. her collaboration with Prof. Wiki, with Prof. Yo, with Prof. Hasima, in all manners of research, shows that mRNA and understanding basic regulatory principles cross-cuts across organisms, cross-cuts across kingdoms, and is applicable to as many diverse facets of science as we can have. And that's where this is such a small world. A small world in where connections are most important, a small world where science can be the unifying factor amongst all the diverse factors that we are trying to address. At the end of the day, we congratulate Professor Jennifer on advancing knowledge in tropical plants that has put Malaysia on the map, and University of Malaya well in the way to being in the top 100. She is definitely a key contributor to our efforts. So she continues to develop her programs. She continues to be an excellent academic, and she continues with the war cry, I suppose, that biologists will one day take over this world. On that note, please join me again to congratulate Professor Jennifer N. Harikrishna in her excellent presentation on a small world of microRNA. Congratulations.
Thank you, Professor Rofina, for chairing this lecture. And thank you, Professor Jennifer, for a wonderful insight into your lifelong work and passion. It's indeed an inspiring story, an inspiring journey, especially for the students and young researchers who wish to make a difference in this world through research. Having said that, ladies and gentlemen, I'd like to invite Professor Jennifer's catalyst, as she calls him, her soulmate, her husband, Dr. Harikrishna, to come on stage to present Professor Jennifer with a beautiful gift. Please welcome. Thank you, Dr. Hari. Next, I'd like to invite Professor Dr. Hashima Nagur, Deputy Director of SEBAR, to present a token of appreciation to Professor Jenny on behalf of SEBAR. Please welcome. Last but not least, I'd like to invite Ms. Lau Su-Yi, representative of Professor Jennifer's lab, to the stage. And we actually have another surprise. We now, with great pleasure, I'd like to invite Yang Berbahagia, Tan Sri Datuk Sri Dr. Salih Muhammad Anur, to come on stage to present Professor Jennifer with a token of appreciation. Please welcome. Thank you very much, Yang Berbahagia Tan Sri, and thank you once again, Professor Jennifer. Ladies and gentlemen, that marks the end of the inaugural lecture. On behalf of University of Malaya, I would like to take this opportunity to thank the management of Faculty of Science, the Corporate Communication Office, students and staff of University of Malaya, as well as all distinguished guests here who have made time to be part of today's inaugural lecture. And apologies for any shortcomings throughout the organization of this lecture. We would like to invite all distinguished guests to have some light refreshments which will be served just outside this auditorium. Ladies and gentlemen, once again, our deepest gratitude for your presence, and have a pleasant evening. Thank you very much. All right, I'd like to call for a photo session. Prof. Jenny with a distinguished guest.